In the name of Allah, the gracious, the most merciful. Very, very good morning, students. Today, we will discuss about the second dominant relation, which is the incomplete dominance. When the phenotype of the heterozygote is intermediate between the phenotypes of two homozygotes, it is known as incomplete or partial dominance. Now, firstly we will write the definition. When the phenotype of the heterozygote is intermediate between the phenotypes of two homozygotes. It is called incomplete dominance. Dominance or partial dominance. Now we will explain the partial dominance by using 4 o'clock plant, which is a flowering plant. So now we will discuss incomplete dominance in 4 o'clock plant. Incomplete dominance in 4 o'clock plant. In 1899, a scientist named as Carl Collins was working with a flowering plant which is called 4 o'clock plant. He crossed the red flowered plants with the white flowered plants. In F1, red flowered plants homozygous red, and white flowered plant which is also homozygous white. When he crossed them, he get all the plants which were pink flowered. The color in its petals is intermediate between the red and white. So 4 o'clock flowering plant. Clock, a flowering plant, is used for working out F1 and F2 by Carl Collins. In this class, all F1 generation is pink which is the intermediate between red and white. And when he self-fertilized F1 offspring, he got F1 multiplied by F1. F1 is self-fertilized. He got the 4 combinations: red flowered plants and two pink flowered plants and 1 white flowered plant in ratio. And so in this case there is no complete dominance, no truly dominance, but it is the intermediate between the two colored flower plants, and there is mixing or blending of phenotypes in this case. But in the complete dominance, there is no mixing or blending of phenotypes. But in this case, he got in F1 generation, the plants were all pink flowered. And so he got phenotypic and genotypic ratios. Now we discuss the ratios in F1 and F2. Ratios in F1 and F2. Ratios in F1 and F2 generation. In case of incomplete dominance, the phenotypic ratio and the genotypic ratio is same. We have already studied in the dominance that the phenotypic ratio and genotypic ratio was different in case of complete dominance, but in this case, 2 types of the ratios are different. Firstly, we see the phenotypic ratio. Phenotypic ratio. It is 1 red, 2 pink and 1 white. One white. This is the phenotypic ratio of the F2 as shown in the Punnett scale. In this scale, we see that 1 plant is red flowered, 2 plants are pink flowered, which is the blending of red and white, and 1 plant is white flowered. And so the phenotypic ratio 1 red, 2 pink and 1 white. And then we will take the genotypic ratio. Genotypic ratio and phenotypic ratio is same in this case. R1 red, R1 R2 pink, 2 pink, 1 red, R1 and R2 and 2 pink. The capital R 1, capital R 2 and 1 white. R2, R2. These are the genotypes of the red, pink and white flowered plants. We see in this case the genotypic ratio and the phenotypic ratio is same, which is 1 into 2 into 1. And so both ratios are same. Both ratios are same. Same in this case. And now we discuss about letter distinction. Letter distinction. In the dominant, in the complete dominant, the dominant allele is denoted, is designated with the capital letter, and recessive allele is represented by the small letter. But in this case, in the case of partial dominance, the letter distinction is different, because in the partial dominance there is blending of phenotypes, but there is no blending of genotypes. And so ratios of the phenotypes and the genotypes are same. The assignment of letter is different in case of partial dominance as compared to complete dominance. And so we will discuss the letter distinction. Letter distinction in case of incomplete dominance. Capital R1 for red color, similarly capital R2 is for white color. Both of the letters are capital, but the numericals are different. For the red flower color, we use R1, and for the white flower color, we use R2. And similarly, capital R1 and capital R2, there is a genotype for red color. Genotype for red. Similarly, R2 is the genotype for white. Because at least 2 genes or 2 alleles represent 1 character. And so these are the genotypes. For the red character, the genotype is capital R1, and for the white colored plants, there is capital R2 and capital R2 and R2. When the gametes are formed in the P1 generation, in the first parental generation, red flowered plants produced the gametes which are of same types. All the gametes contain R1 allele, and similarly R1 is the homozygote, and this is the white flowered plant and the genotype is capital R2 and capital R2, and in gametes only one of the two alleles is present, which is the only R2, and all types of the gametes are same in both cases. And so when these gametes unite together, we get F1 offspring, which is R1, R2, and so phenotypically it is pink. There is blending in case of phenotype. But when he self-fertilized F1 offspring, he get the three types of the genotypes R1, R1, which is red flowered plants, R1, R2, pink flowered plants, R1, R2, pink flowered plants, two are pink flowered plants, and one is white flowered plant. In F1, R1 and R2, they are segregated when gametes are formed during meiosis in the F1 progeny, and so there is no blending at genetic level. Now, the next point is blending not occur at blending not occur at genetic level. At genetic level, alleles are segregated from each other. Alleles are segregated from each other when F1 offspring form gametes. There is no blending of alleles R1, R2. They are segregated when meiosis occurs, when there is gamete formation. And so capital R1 in one type of gamete, and R2 is in other type of gamete. So two types of gametes are formed, and so these alleles are segregated again. In this case, these are the male alleles. When we cross F1 and F1, R1 and R2 are the male alleles. R1 and R2 are the female alleles. And so, blending does not take place at genetic level, but blending takes place at phenotypic level. And the next point is, there is no need of test cross. No need of test cross. No need of test cross. Because the phenotypic ratio and the genotypic ratio in case of incomplete dominance is same, and so we do not need any test cross. The phenotypic ratio and genotypic ratio is same. Is same. Same. Phenotypic ratio is 1 into 2 into 1, 1 red, 2 pink and 1 white, and genotypic ratio is also same. Capital R 1, capital R 1, and 2 plots are R1 and R2, and one plant is white, which is R2. In this case, the ratio is again same. 1 into 2 into 1. And so, the genotypic ratio and phenotypic ratio is same. So, we have no need of the test class. But in case of dominance, we have studied that the phenotypic ratio and the genotypic ratio was different. And so, in that case, we needed the test class. Mental laws or principles are valid in this case also, because there is separation and segregation of allele when gametes are formed at any level, in P1 as well as in F1. When gametes are formed due to meiosis, the alleles are separated and segregated. There is no mixing of alleles, there is no blending of alleles, there is no blending at genetic level. And so we can say that the mental laws are also applicable in this case. This is the explanation of the incomplete dominance or partial dominance. In this case, we got pink flowered plants in F1. There is intermediate phenotype in F1 plants, which is pink. And thank you very much.